for many years in my lab, and it's work done by a group of very talented collaborators that I've had the pleasure to have in the lab, like Klaus Wimmer, Joao Barbosa, Heike Stein, David Bestue, and the work of them is what I'm going to be presenting and putting in context here. So what I want to tell you about is how we have been working towards making computational models useful in our institution. This is IDIPAPS. This is a biomedical institute, and we've been pushed to make computational models interesting or bring in insights into clinical practice. That's a hard. So this is it's not biomedical. It's a clinical institute associated with a hospital. So the way we've been thinking about that over these years is that we're comfortable with our computational models working at the level of neural circuits. Now they're asking us how that could contribute to making some insightful findings about diseases. And we figured that a good way to go about it is to think about behavior. We can link neural circuits to behavior. We can link disease to behavior. And computational models can be a very useful tool to actually try to make consistent, to bring in a consistent understanding of how all these three pieces, these puzzle pieces come together. So we've done this in a very specific domain, and that's what I want to present to you today. And that's by looking at a specific behavior, and that's working memory, and showing how a Bump-attractor model is able to reproduce many non-trivial features of behavior that occurs in a specific memory task. And I'm going to be focusing all the time in one task, and let me show you first this close match between the model and the behavior, and then I'm going to work through the talk and reach to the other puzzle pieces that we have formulated here. So what is this behavior? This is a visuospatial delayed response task, very, very simple task, but still it comes with a set of interesting behavioral readouts that we can simultaneously record in a data set. I'm going to show you a specific results from one data set that Heike Stein collected in the lab in humans. We asked people to stay in front of sit in front of a screen and fixate their eyes on a central dot. Then a cue appeared peripherally, briefly flashed, it went away, and then there was a delay that could have varying lengths, 0, 1, and 3 seconds. After this delay that occurred randomly in randomly interleaved trials, the probe changed color, and then participants had to move this dot to the location where they remembered the cue having appeared. What we record is the exact location they report their memory to have the memorized location of the cue, and then we compute the error as the angular distance between this clicked location and the actual location of the stimulus. Over 22 subjects, here is the distribution of errors that we get, and it's nicely peaked, looks like possibly like a Gaussian, reflecting that people were doing the task essentially. Now, if we look at this distribution of errors that we obtain in this task, we can see that they're not all random errors. There are a number of systematic errors that we can tease apart and analyze independently. So one of the errors that we can observe, one of the sources of errors that we observe is when we plot these errors as a function of the target. So where the target was presented here on the x-axis, and where the error that people made. And what you can see, if we zoom in here a bit, is that these errors are not uniformly distributed. So there is a systematic bias that occurs that is marked by the location of the cardinal axis. 0 would be horizontal, 90 is vertical. So you see that at those cardinal locations, there is a specific pattern of errors. If you think about it, these errors reflect a cardinal repulsion. So for instance, if we go to one side of the vertical meridian, and here I'm plotting in red the location of the cue, and in white you have all the endpoints of saccades when the location was that one. what you see is the saccades are biased away from this vertical meridian. If you just go across the meridian, you see the reverse effect happening there too. So there's repulsion from both sides of the meridian. If we go to the horizontal meridian, then we get also these effects. So that we have repulsion at one side, repulsion at the other side. So this cardinal repulsion is very prominent. It's something that we see systematically in this behavior, and we can remove it from the data by modeling our errors as in a linear model where we include a sawtooth regressor that has this shape. We can estimate the amount of cardinal repulsion that we observe in each participant, and we can remove that as systematic bias. When we do that, we can then analyze residuals. Residuals tell us what is the rest of errors that we can look at here. And there's still one further systematic error that we can observe when we plot those errors as a function of where the current item is in this trial relative to where the trial in the previous item was. And that's the x-axis here. And here again, we plot the errors. So that is again not flat. It's a much smaller bias, as you can see here. But if we zoom in, you're going to see that there is a characteristic shape, and that is this positive lobe for relatively close locations between the previous and the current item. That means attraction. So we go to some of this. Now you see that instead of being repelled to the meridian, this specific case, the saccades are attracted to the yellow dot, which is the item in the previous trial. That's what we observe here in this location, in these examples over here. So there is an attraction of responses towards the previous trial. A slight effect. This is not really a change to the previous item. It's just a small bias. Again, we can analyze that. We can remove that by running a linear model with this basis function, with this shape, and we can estimate those serial bias, the strength of the serial biases, and remove them. And then the residuals of that second step of regression are another distribution of errors here that now is better aligned to zero, is more centered, looks more Gaussian, and we attribute that really to random Gaussian distributed errors, and that's what we're going to call errors, precision errors. So we have two sources of systematic biases, cardinal repulsion, serial biases, and one source of random errors that we can extract for each participant given their responses in one single task. We can get these three possibly independent measurements. Now, importantly, these, yes?
Can you just minimize my video so we'll see your titles?
Oh, yeah. Let me.
Just move me somewhere. Yeah, perfect.
Okay. Okay, so again, this is when we look at these three different sources of errors in this data set, and now we analyze separately trials in which the delay was 0, 1, or 3 seconds. What we see is that the three of them change with the delay, and that's an important feature. What that tells us is that these errors, or these different sources of errors they are built in, or they are accumulated in the circuit, whichever circuit is responsible for maintaining information in working memory, because the encoding part of the trial is the same in the three sorts of trials. The motor component is also equally engaged in all the trials, and the only thing that differs from these three different sources of trials is how long the thing is in the delay. So we think that this is an important feature that makes these three sources of errors important markers of what goes on in whatever circuit is doing this memory. Now, this has been shown in many different, so these different biases and errors have been reported for many other tasks in space, like here, but also in orientation or in color. And so we are just showing how here I'm just showing you how we can see this in one single data set all at once, and making the point about this delay dependency. So we have a way, as I was telling you, that to model this using a Bump-attractor model. This is a model that has a long history in computational neuroscience. It was first proposed in the 70s by Wilson Cohen and Mary. And it's evolved from there just by making it more biologically realistic and by understanding better the different components of this model. Here, I'm going to show you simulations from one like this that incorporates a few additional features. One of them is that, so essentially, remember, in a ring model, what we have is excitatory neurons and inhibitory neurons. That's what I'm going to present today. Is a model with excitation and inhibition, and excitation is tuned so that excitatory neurons that are close on the rings, that means they prefer similar stimuli. They are more strongly connected than neurons that are further apart. Inhibition has a weaker tuning in their connectivity, but it's very important because it constrains activity in the circuit to be localized. What we add in this classic ring model is, so what we're gonna put in is synaptic receptors. So this will be a spiking model, and it will include some receptors and MDA and AMPA. And what we will do is to include in these receptors short-term plasticity. So we have mechanisms of facilitation and depression, and we're going to tune them so that facilitation is dominant. So that's one element that we have additional to the classic ring model. And then in addition, the other thing we have is that this connectivity is not going to be translationally invariant. It will have just tiny bit inhomogeneities that are going to be priming or marking the cardinal locations in a specific way that I'll come back to later in the talk. So when we hook up neurons in this way and we build a network with all these mechanisms, we can have a Bump-attractor, and this is just a simulation showing that there's a bump is introduced by external current. Now the current is released, and then the activity remains localized in the circuit by virtue of this recurrent connectivity that produces this reverberation and the inhibition that constrains the activity to remain localized. So at the end of this delay, we can go back to the simulation and extract the peak of this the peak of this distribution. This would be the distribution of spike counts at the end of the delay across all neurons. And if we get like the center of mass of this distribution, we are picking the memory, what the network is telling us that the information was what information was introduced at the time of the cue. So that would be the readout, and we can get sort of behavior from this network. So this is what we do essentially, is to run a population vector decoder through the simulation, and that gives us a decoded angle that is usually random, except when there is some pattern stored in the circuit, in which case it takes like the center of mass. And here, so that would be like the instantaneous readout of the memory through the trial. We picked instantaneous readout at the end of the delay as output of a simulation. So I'm going to be plotting this center of masses through the talk to represent multiple simulations that we have run. So when we run thousands of these network simulations with different delay times, and then we analyze these responses, these outputs at the end of the delay, in the same way that we analyze behavioral data from the human experiment, we obtain something very similar to what it showed you. So we get a distribution of errors, a pattern of cardinal repulsion and serial bias that is delay dependent in the same way that it was for human participants. So this is something that we've been studying and many other labs have been studying in different ways, both for precision, cardinal repulsion, and serial bias in computational models, showing that they can emerge as a result of synaptic plasticity, cardinal repulsions in inhomogeneities in the connectivity, and precision as an accumulation of the. Yes? Okay. So yeah, in this way, I showed how this bump-attractor model, just one model, can pick and model accurately these three sources of errors and biases that are readily obtained from this behavior in human participants. Now, a big effort in my lab has been to show how the predictions from this bump-attractor model are actually supported by neurophysiological recordings in the prefrontal cortex of monkeys when they do this task. And that is a way to give plausibility to this hypothesis that is inherent in this modeling of effort, that this behavior relies on the activity of a microcircuit. So it's the action of the microcircuit what is generating these different sources of biases. So let me show the evidence that supports that by showing you how that bump-attractor model actually also connects pretty tightly to neurophysiology obtained at the microcircuit level in the monkey. So these are recordings. This is a collaboration with Christos Constantinidis that I have a long time collaboration with. In monkeys that are doing this task, the only difference is instead of a click with a mouse, what they do is a saccade eye movement to report the remembered location. And we obtained data from the prefrontal cortex. Christos obtained data from area 46, area 8, 9 from the prefrontal cortex of the monkey, and we took those neurons that are related to, or that could be part of this computational model, or the bump-attractor in the prefrontal cortex, because they had tuned persistent activities. Essentially, when we look at those 204 neurons out of the 822, those have tuned selectivity as defined in the Q that persists through the delay, as is represented here. So we're going to look at those neurons that are part of 94 ensembles in which there were a few simultaneously recorded cells. So I'm going to go through each of these three different effects and show you one prediction that is verified in the data. We published for two of the effects, for the precision and the serial bias, we published two whole papers in which we look at many different things. But I'm just going to, for sake of time, just going to present one prediction per effect. So now let's first think about the diffusion. That's the distribution of errors that grows as the delay becomes longer. So the way that the model does that, and that's been proposed for a long time, is that as the here I'm plotting many different simulations when the Q was presented in the middle, and what you see is that as the delay progresses, the center of mass of those different bumps in the simulation drifts away from the original location, or diffuses away rather, and the distribution of endpoints at different times during the delay grows and becomes broader and broader. And that's the way that our simulations can reproduce the behavioral effect, because of this accumulation of noise. Noise is making those bumps, internal noise in the circuit, is accumulated in different ways through different trials, so that the bump in some trials moves to one side or to the other. Eventually, at the end of the delay, the endpoints, the center of those bumps, have drifted away from one another. And that makes this distribution grow. So what can we predict? When our recordings are not looking at the full, or are not able to obtain the full view of what the network is doing, but just one single cell can be recorded. So I'm going to show you here one trial first, and then another trial. And this would be like the population activity in black. And now we're going to be looking at this one cell in red. And this yellow triangle is marking what the decoded variable is at that point. So where, if we would stop at that point, where behavior would be going in that one trial, right? So if we run this simulation, what you get, well, this is not really a simulation, this is just a schematic. What you see is that in this particular trial, behavior will be more, say, to a more negative angle than where it started. It started at 0. Now behavior would would say that the item was at -50, and this neuron fires more. If we do the other thing, another trial, what you're going to see is that now in this other trial, the behavior goes to the positive angle, so away from the preferred location of this neuron, and this neuron fires less. And if you think carefully about this, this is something that we should predict overall. So when a neuron in a given trial, on a trial-by-trial basis, fires below its mean firing rate, that if you think a bit about the underlying population, that must mean that the bump has moved away from the neuron preferred location. Wherever it was, now it fires less. Instead, if it fires more, it means that the bump has moved closer to the preferred location of the neuron. So we predict this correlation between the trial-by-trial fluctuations relative to the mean of each neuron and deviations in the monkey behavior towards the preferred location of the neuron. And that's exactly what we see in the model. So we analyze data in the model looking at single cells and correlating single cell fluctuations of firing rate relative to the endpoint behavior. What we see is that the correlation grows through the delay, as the intuition that we got here, and that is in particular dependent on where we present the stimulus relative to the preferred location of the neuron. If the stimulus is close to the preferred location of the neuron, very quickly in the delay, that bump is gonna make the neuron change firing rate, and we're gonna see a strong correlation grow up quickly. Instead, if the stimulus is presented far from the neuron's preferred location, then we predict that this increase in correlation will take longer, because the bump will take longer to reach and engage the neuron in a different firing rate. So that's exactly what we tested in this prefrontal data. So we extracted spike counts in different time windows through the delay, and then we correlated that with the behavior of the monkey, whether it made a behavior more clockwise or counterclockwise, and in the direction of the neuron preferred location or away. And we correlated that, and what we saw is that this correlation grew and became positive in the middle of the delay, and that this happened early when we considered stimuli that were placed close to the neuron's preferred location as compared to stimuli that were further away from the neuron's preferred location, just as we predict with the model, and which gives the idea that there is something evolving in the delay that is engaging neurons progressively in the form which is consistent with this bump moving about in the circuit. Okay, so that was one of the predictions, so one of the effects. The other effect that I told you about was this cardinal repulsion in the model. As I told you before, we introduced heterogeneities in the circuit instead of having a perfectly translationally invariant connectivity pattern. What we had just a tiny bit inhomogeneity, and here you see it. If I zoom in, so it's really small. It's not it's a small modulation of this otherwise perfect symmetry, but this small modulation that is priming specifically the cardinal locations is what causes these cardinal biases. Essentially, here I'm showing you simulations with uniformly distributed cues. It's uniformly at the beginning, but as the delay progresses, you see that this uniformity is raised, and we see stronger and stronger biases towards these cardinal locations. And that's what we end up seeing, you know, in the simulation. If we take delay 0, there's no bias. At delay 1, we find this cardinal repulsion bias. At delay 3, those have grown and they're bigger. Okay, so there's also prediction we can make about that. Suppose you know, you have to think about the tuning curve. Again, we have single cells, so we can look at these tuning curves. But how do we build a tuning curve? A tuning curve is built by averaging across trials. Now, in our framework, in a given trial, even if we present the same stimulus, and this would be the result of a stimulus presented at 180 degrees, I mean, for this peak here, this would be, this point over here is 180 degrees, but in fact, you have to think of the different trials having a spread because you know those bumps are not perfectly pinned on the same location. So the true bump shape is going to be when we average across trials is going to average out the true bump shape, and the tuning curve is a smeared out version of the true pattern of activity. That's what I'm plotting here. So this in blue we have the tuning curve. In dashed line, this would be the underlying bump shape, and these dashed lines are these different initial conditions of the positions when we present the same stimuli here at the center. Now, if we consider cardinal neuron and oblique neuron, and we take this as being at the beginning of the delay, at the end of the delay, these central locations of the encoded variables that are there at the beginning will have evolved differently for the two neurons. For cardinal neurons, they will have been repelled from the cardinal, so they're going to move away. For the oblique neurons, instead, they're going to be attracted. So what that means is that the smearing will be stronger for cardinal neurons as opposed to oblique neurons, where it will be actually diminished. So we can get tuning curves that are narrower for oblique neurons as delay progresses compared to cardinal neurons that get broader tuning curves. This is what we see in the computational model. So when we analyze the data in the computational model, separating neurons that are in the cardinal versus the oblique direction, we take the tuning curves for different presentations of stimulus in different locations. What we do get is that the width of the tuning curves evolves in the delay as predicted by this intuition. This is work, by the way, is done by David Bestouet in the lab, and it's still unpublished. When we look at the prefrontal cortex neurons, we see the same thing. So we look at this, it's again the same 204 neurons in which we saw before traces for the diffusion of the bump. Now we'll look at the tuning curves when separating neurons that prefer cardinal versus neurons that prefer oblique locations, and we see that the same predicted evolution of tuning curve widths occurs during the delay as our model predicts, which suggests that these dynamics of the dynamics that in our computational model is responsible for cardinal repulsion could be also in play in this one microcircuit in the prefrontal cortex. And finally, let me go to serial biases in the model. This is work done by Joao Barbosa and Heike Stein in the lab, and it's also published, and we have many different ways of looking at this, but let me show you one signature of the model that is to be seen in this 204 neurons again. So remember, we have short-term plasticity in these synapses, and by virtue of that, when we simulate two consecutive trials, as in this one simulation, what we find is that the second trial is not completely independent of the first, because there is this calcium signal that is supporting short-term facilitation that bridges the gap, the intertrial interval between the two trials, and makes the bump that appears in the second trial to feel an inhomogeneity in the circuit that depends on the previous trial. So that if they are close enough, this bump is going to feel attracted to this synaptic trace that remains in the circuit, and that's what we see in serial. That's the source of these serial biases in our network simulations. So what can we predict about this that we can test in the physiology data? So one feature of this sort of synaptic memory that has been reported in this paper by Mongillo, Barak, and Sodix, is that the memories that are kept in this synaptic substrate can be reactivated, and that happens when you introduce a non-pattern input. You sort of excite all excitatory neurons in the circuit, and because this network is close to the Bump-attractor condition, so it's going to want to reactivate with a bump, and that's going to happen more often than not at the location where the synapses have been primed by this facilitation in the previous trial. So it tends to, this reactivation tends to occur at the time of the previous trial. So when the new comes in, this reactivation enhances the attraction that was seen without the reactivation. That's what we tested in simulations. So without reactivation, we have a pattern of attraction, which gets greatly enhanced when we reactivate in trials in which we reactivate. So we figured that could be going on in this circuit. So we looked at the neurons in these 204 neurons, and now I'm plotting you centering our interest on the intertrial interval. So we have the previous trial here, the next trial in this side. This is the intertrial interval, and what I'm plotting here is the decoded, the decoding from this subset of from these ensembles of neurons that we have of the previous item. So because we select the neurons for being tuned, this is no surprise. We had to have good tuning in the previous trial. But then the interesting thing is what happens after that. And what we see is that the decoding decays, it disappears in the intertrial interval, but then there is this period of reactivation that we, or re-decoding that we observe right before the next cue appears. This is interesting, is reminiscent of what of this reactivation generated in this synaptic memory. And we asked whether this had indeed to do with the strength of the serial biases in the next trial. In order to test that, what we did was we selected trials based on the physiology, without looking at behavior, only looking at the physiology. In a given trial, we decoded the cue represented at this time point. If that cue is close enough to the previous cue in the previous trial, we put those in the back, that trial in the back of the reactivation or putative reactivation trials. And if that decoded angle is very far from the previous trial, then we put it in the back of the non-reactivated trials. And then once we have two backs, one with trials with good reactivation, one with trials without reactivation, then we go to behavior in the monkey, and then we ask, are there more biases in those trials in which reactivation was more probable? And indeed, that's what we found. So this green would be the reactivation trials, in black the no reactivation trials in the data, and this is the prediction from the model. And you can see that indeed we found that the reactivation, the serial bias was related to those more accurate representations of the previous item right before the cue appears, suggesting again that this reactivation is responsible for, or is partly responsible for the serial biases observed in the monkeys. So yeah, with this, I hope I convinced you that the Bump-attractor model not only reproduces the three features of this behavior quite closely, but it also does this in close correspondence with what we see at the level of the microcircuit and prefrontal cortex, suggesting that this behavior is mapping or is measuring microcircuit function in a relatively, well, through three different measurements that can be telling about different mechanisms. And that's where we thought we could, about these three different mechanisms, which are specifically, so when we think about precision of the memory reports, we are talking about, noise in the circuit being accumulated by this bump that moves about, and that will depend on structure of noise correlations. There's many works on that that have thought about that. Cardinal repulsion would be about inhomogeneities in the line attractor. And serialized in working memory might be reflecting some slow activity-dependent mechanisms. We propose short-term facilitation, but there could be others, as long as they have some slow time constant that is able to bridge the intertrial interval and is activity-dependent. Okay, so we figured that, yeah, that's how we thought about this behavioral readout being useful to test in neuropsychiatric disorders, because our model is telling us how it could relate to different separate mechanisms in the microcircuit. So we convinced medical doctors in our institution to to start a behavioral experiment in participants, including 2 clinical populations. This is work done. The leader was Heike Stein, who did a tremendous work here, coordinating all this work with neurologist Dr. Dalmau and Salta Maria, and psychiatrist Dr. Castro. We took advantage of the fact that Dr. Dalmau discovered this disease, anti-NMDA receptor encephalitis. It's an autoimmune disease that attacks NMDA receptors, so that these people have like a sudden internalization of NMDA receptors that are removed from the central nervous system. And interestingly, that causes initially symptoms that very much resemble schizophrenia. So that's why it's been a model of interest also in the context of schizophrenia. And we are in a unique position to be able to study that, because Dr. Dalmau is here, and all these patients, which is a rare disease, so we have to invite them and make them travel over to Barcelona. So they are very, very much willing to see Dr. Dalmau. So it's easy to recruit them and have these studies. So over 3 years, we've been able to record about 20 patients. So we've increased that by now. And we tested them in this behavior, thinking about measuring those 3 different outcomes, and then making prediction or interpreting that in terms of dysfunction at the microcircle level. So essentially, when we looked at precision, this would be the one thing that we looked, and that's where we thought initially that we would see a difference, because there are reports in the literature that attribute changes in precision in this task in schizophrenia patients, but they typically go to much longer delays. So when in our hands, we didn't see differences between control schizophrenia and encephalitis groups in terms of the errors that they make at different delays. They more or less are the same. So that part of our readout doesn't seem to be affected in these 2 groups. When we looked at the cardinal repulsion, we also did not see any difference. So we see cardinal repulsions pretty much equivalent for the 3 groups, and they grow with delay similarly. So that doesn't seem to be affected either in these 2 diseases. But then when we looked at the serial bias, the surprise was that we saw tremendous effect of really strong modulation in the diseases. So essentially, and more interestingly, this was not observed when we have 0 seconds delay trials. So trials for which there is no delay, there's the same motor requirements, the same encoding requirements as the other trials, but there's no memory. And then the 3 participants, the 3 groups, are essentially indistinguishable. But as the delay grows longer, what we see is that the attractive biases that were observed in control participants are totally abolished in schizophrenia, which actually shows repulsion, and very much reduced in encephalitis. So this is a remarkable finding, I think, because there's not much biomarkers at this low psychophysical level that discriminate this well between neuropsychiatric diseases and controls. We model this, and this is work, again, in this paper by Heike Stein and Joao Barbosa. We modeled this with our model, and we tried to see whether what manipulation in the model could reproduce this, and we tried to play with NMDA receptors in excitatory neurons and inhibitory neurons, or directly with the short-term plasticity mechanism that we incorporated. And what we found is that we could not find the same pattern of behavior unless we reduced significantly the amount of short-term plasticity that was included in the model. So essentially, this parameter that produces this gradual change that mimics our results is a reduction in short-term plasticity. So yeah, sorry. Based on this, so putting together all these pieces, it looks like there would be a microcircuit dysfunction in these 2 diseases, but it's not widespread. It doesn't affect. So if there is an effect at the level of the excitation inhibition ratio, it would probably mean changes at the level of the diffusion or the other or cardinal repulsion, which we don't see. Instead, it seems like this manipulation. So we're proposing that an alteration, a specific alteration at the level of short-term plasticity could be happening in these microcircuits. In particular, this is, I have to say, stabilized schizophrenia. So it's not just the full spectrum of schizophrenia phenomenology. It's this stabilized condition might be characterized by this short-term plasticity reduction. So one prediction of this model, again, is that if the short-term plasticity is absent, then these reactivations should not occur. And we tested that by measuring EEG in these participants. In control participants, we decode the location of the previous item from the EEG. It's strong during the delay. It's absent in the intertrial interval. It comes back before the next trial, just as in monkeys. So we also have reactivation in control participants. But when we look at the patients, that reactivation is gone. And the correlation with behavior that we see in controls, so participants that have more reactivation also have more serial biases, that is not observed in the participants. So that is also some a result in the humans that supports the idea that there is a change at the level of short-term plasticity in this participant or short-term mechanisms. So this would be a summary of what I told you. So the Bump-attractor model links very closely this working memory behavior in this one task and the cortical microcircuit dynamics. And we reached in one specific point towards neuropsychiatric disorders, and with a specific experiment that we did in our institution that allows us to identify a specificity in the alterations that occur in this disease at the level of the microcircuit. And yeah, I just would like to thank everybody that participated here, especially people that did all the work: Joao Barbosa, Heike Stein, Klaus Wimmer, and David Bestouet. They worked in all these different papers and to connect closely the model with the neural data, and then Heike Stein and all the medical doctors in the institution that made this possible. And with that, I just want to thank you, and